Hi everybody. Today we are going to study about epigenetics and stem cells. As usual, here are some references from Wikipedia. Some terms sounds like quite technical, but we are going to learn what they mean and why they are important for this study. So for details, we are going to study, in comparison to conventional genetics, what is epigenetics? And then, what is the CpG island and its role in gene expression? And two technical terms, so methylated DNA immunoprecipitation and chromatin immunoprecipitation. And then moving to stem cell and its application. And not only to understand what is a stem cell, we are going to learn how to make stem cells. Okay, let's start with the epigenetics. Today we are going to see one nice intro movie first, and then moving into the details of epigenetics.
Meet Lucky Lyle and Trouble Tim. Now you might think they look a lot alike. That's because they're identical twins. But imagine them being raised in completely different environments. As adults, they turn out very differently. Lucky Lyle is a model citizen. He goes to work, pays his taxes, treats others well, and gets an education. Trouble Tim is on a different path. He robs banks, doesn't pay taxes, and gets his first prison sentence at the same time as Lyle gets his diploma. Their genes are identical, so it must be their separate environments that have made them so different, right? Well, there's more to it than that. Much more. We need to rewind a bit in time. You see, one of the great questions of humanity is, what makes us us? Early explanations had a hint of the supernatural about them. As the centuries whizzed by, science emerged and fought a tough battle to gain credibility. Eventually, social science emerged with the popular theory that our environment is what makes us us. Let's call this nurture. Undaunted, the biological sciences presented another theory: genes, an unchangeable blueprint in you from birth, decides everything. Let's call this one nature. So, are we a product of our genes or of our environment? The battle raged on. Observations in a newer field of study suggest that both are partly right. This field is called epigenetics. It means above genetics and has to do with how nature and nurture interact. At the heart of this discussion is one simple question asked by researcher Michael Meany: what makes a good rat mama? Well, for a rat mother, nothing says love and care like some nice, vigorous licking. The higher the number of licks, the more love and the better the mama. With this in mind, the researchers compared two groups of rat mothers, one that licked their babies a lot and another that didn't. They found that when babies grew up, they carried their mama's behavior with them and passed it on to the next generation and the next one, and so on. But could it simply be that this particular group of rats was genetically predisposed to produce caring mothers? Well, to test this, the researchers took newborn babies from low-licking mothers and fostered them with high-licking mothers and vice versa. It turns out that if the mother is a high licker, the rat baby becomes a high licker too, regardless of whether it's genetically related to her or not. So genes really don't have anything to do with it then? In true scientific fashion, the researchers dove right into the brains of these rats to see what, if anything, was different in the brains of the babies of high-licking versus low-licking mothers. When they looked at genes that possibly play a role in motherly care, they found crucial differences. Newborn rat babies have clusters of molecules called methyl groups attached to these genes. These methyl groups silence the gene, effectively switching it off. The researchers discovered that while the methyl groups in rats from low-licking mothers were still attached, in the rats from high-licking mothers, these methyl groups had disappeared. This was also true for the rats that had been adopted by high-licking rat mothers. The care these rats received from their mothers actually physically altered their genetic expression.
How about it? Did you get a good idea what epigenetics means? To summarize epigenetics, let's compare with the Mendelian genetics. Mendel started that conventional genetics are the basic principle. So in his definition, let's say phenotype and genotype is very closely related. If you see any kind of phenotypic change, then that means somehow genotype is also changed. And genotype is digital information. That's why that change means somehow the sequence of gene is changed. Those sequence change we call mutation. So this is the basis of conventional or Mendelian genetics. Now we are in a new situation that the phenotype is changed, but that change is not reflected in genotype. Then using conventional genetics, we cannot explain this new situation. So now we have the view of genetics. That new are the genetics we call epigenetics. Epi means above. So that means above conventional genetics, we need to have a different explanation, different understanding. So that's epigenetics. To move on, let's think about the genetic the expression structure. When the gene is expressed, first of all, there is DNA binding domain should come first, and then bind this area, so-called promoter area. And then this recruit the activation domains, including RNA polymerases. But this is a very, very simplified version of the gene expression. Once this binding happens, then we said gene expression started. The real situation is very, very complicated, something like this. Now, first of all, you can see here, this is a Tata box. This is a part of a promoter sequence. Not only this promoter sequence, we have another enhancer sequence or silencer sequence. And in the previous pictures, we showed only one activator, but it can be multiple activators. And sometimes not only activators, controlling repressor can bind here too. Then activator-repressor balance is important, whether to start or not to start of gene expression. And eventually everything is balanced out. Now DNA polymerase can start that gene expressions. That means in this genetic sequence, nothing is changed. Still, they can affect whether the gene can be expressed or not. In the previous movie, there was some small clues. Methylation on DNAs. If a methylation happens, somehow it can suppress gene expressions. Some clues there. Now let's look at DNA structure. DNA is composed of A, T, C, G, and RNA has a very similar structure, but T and U is the difference. Watson and Crick, the hydrogen bonding site is exactly the same. Only difference here is one extra methyl group in T, not in U. Now, another methylation for epigenetics happens on C, cytosine. Now if you look at this structure, cytosine 5 position is methylated here. Now the relationship between this methylated cytosine to natural cytosine is very similar to T to U, isn't it? If you look at Watson and Crick base pairing, C and G is interacting through 3 hydrogen bonding here. And methylation position, do you remember on C? That position should be here. Then if methylation happens here, is that in the site of minor growth or major growth? Can you recognize? Ribose is connected here. Then, yes, this is minor growth and this is a major growth. Do you remember? So that means even methylation happens here. The methylation site is located to the open space here. It does not interfere with the Watson and Crick base pairing. That's the important part. We have 4 base pairs, ATCG. If every C can be methylated, average percentage can be about 25%. Then maybe the regulating role of methylation may be a little difficult to utilize. So the methylation can happen only on the sequence of CG. It may be a little difficult to pronounce as a CG, so that's why utilizing one phosphate bond in between, we call this linkage as CPG. And this CPG, if you think about the opposite complementary sequence, if you read it this way, CPG, and then complementary sequence also come as a CPG. So this kind of relationship we call is a palindromic sequences. Be careful, CPG and GPC is not same. You understand? If it is only one sequence as CPG, how many percent we can expect this sequence to come up? The total combination can be 4 by 4, it's a 16 combination. Then the probability of this CPG among total 16 different combinations, it should be 1 over 16. That's approximately 6%. The CPG sequence distribution is not even. In normal area of DNA, the CPG count is not so high. But the CPG sequence is highly localized in 5' area of genes. 5' is upstream of gene. That means when this CPG is methylated, then the gene expression can be suppressed. That also means that this 5' is quite important to decide the gene expression regulations. So that's why if DNA is methylated, the general phenomena is the transcription is suppressed. So that's what we call transcriptional silencing. It may have a lot of implications, and some of them can be tissue-specific gene expressions, and X-inactivation, and genomic imprinting. Let's look at the first case of a tissue-specific gene expression. We have studied about transcriptome. Every cell in our body has exactly the same genome in principle, but different tissue has a different expression of those genes. Human genome has a little more than 20,000 different genes, right? Then we can think the scenario, in the beginning, we expressed all the genes. And when we started our life, we were one fertilized egg cells. And then dividing those cells and make more part of those body, and then some part of the body should have specific functions and specific roles. Then one mechanism to control those selectivity, maybe we can block out some part of those genes by methylations. Then different cells in different tissues, still they have exactly the same sequence of ATCG. That means the genome sequence is still exactly the same. Only some part of those cytosine can be methylated, then those genes cannot be expressed. That's how different tissue can make a different set of mRNA. And from different mRNA, we can make a cDNA. Those cDNA library, they are tissue-specific collections of expressing gene sets. And that's why this transcriptome, in other terms, we can call epigenome. Even identical twins who has exactly same genomic sequences, if their DNA methylation pattern is different, the expression pattern can be changed. That was the example we have seen in the movie. If you look at the tissue-specific gene expression, we can find that different tissues has a different pattern of methylations. In this picture, this is a different tissues, and then yellow color is unmethylated, and blue color is methylated. Maybe early stage we have a lot of genes are yellow, that means a lot of genes are expressed. When our body is developed further and further, now we can see a lot of blue regions are coming up. That means block by block, those genes can be removed from gene expression and only express some of those yellow color area. Those can specify the property of each tissues. Here we can see some area of genes is always turned on from the early days and to the end days. Now these genes can be maybe very basic functions, maybe housekeeping genes. And some area here highly variable among tissues, and these genes can define the unique property of different tissues. Now can you look at the human chromosome? And by looking at this human chromosome, can you tell whether this individual is a male or female? In human chromosome, we have 23 pairs of a chromosome. Now, only difference between male and female is these two. If you have 2 X, then it's female. And if you have 1 X and 1 Y, it makes male. Then this individual should be male, isn't it? The size of X chromosome is much bigger than Y chromosome. If a female expresses both of those X chromosomes, the power of X chromosome is dominating over male, and it can be quite difficult to balance out. That's why in female case, even though they have two X chromosome, interestingly, one of those X chromosome is inactivated by so-called X-inactivation. And female has two X chromosome in every cells, but one of those X chromosome can be condensed and deactivated. The key mechanism of this deactivation is done by DNA methylations. If you look at any female cells, they should have two copies of X chromosome, but one is condensed form and inactivated form. Only the other one is normal and activated forms. Maybe the next question is, which one is inactivated? Seems like very interestingly, it's a random choice. So depending on those different tissue or different cell, they choose randomly one X chromosome. It doesn't matter from mother side or father side, inactivate one of them. And the genetic impact from father side and mother side, at least for X chromosome, is averaged out. Now let's look at the DNA methylation pattern change over whole life cycle. When a sperm cell meets egg cell, it forms one fertilized egg cells. This is our starting point of our life. Then the cells are dividing and it reaches in blastocyst stages. During this process, most of those DNA methylation pattern is removed. That means in blastocyst stages, as most of the genes are activated, almost every genes make proteins. Then when it is differentiated into different tissues, now different tissue start to have a unique methylation pattern and then make unique property of different tissues. Small population of cells are reserved for next generations to make sperm cell or oocytes. Now during this process, they have very unique methylation pattern is rebuilt in these stages. When they meet to make a next generation of fertilized egg cells, now they have quite a unique methylation pattern depending on whether the genes are from sperm or from oocyte. Most of those DNA methylation pattern is removed during this development, but interestingly, some genes they stay in their methylation pattern and even they can be transferred to the next generation, to the sperm cells and oocyte. Let's think about this strange phenomena. In Mendelian genetics, if we have a gene A and B, now A and B from sperm cell and from egg cells, they meet together and they can distribute evenly of those AB gene into next generations, either male baby or female baby. In this case, the sperm cell, let's say, they carries B gene with methylation, and egg cell, they carries A gene with methylations. And when they make a male baby can make sperm, and female baby make egg cell. Now, sperm cells, all the sperm cells, the gene methylation pattern is following exactly the fathers. It ignores the methylation pattern of mothers. So that means even though those gene distribution, genetic sequence distribution is following Mendelian rules, but those methylation pattern is follows only the fathers in this case for B gene. For egg cells, it follows the mother's pattern. That means this A gene's methylation pattern is transferred only to female from mother to daughter direction, and then B gene methylation pattern follows from father to son directions. And this phenomena is called as a genomic imprinting, and this is utilized as the evidence of competition between sexes. Now let's think about the chemical method how to determine the methylation site of a cytosine. Interesting chemistry has been developed for this, utilizing bisulfite. If you add bisulfite to the cytosine, bisulfite can introduce this sulfate group to the cytosine, and eventually it can change cytosine into uracil. Now, if the methylation happens here, this reaction becomes much slower, so methylated site can be protected from these bisulfite reactions. So that means if you have two different sites, methylated cytosine versus unmethylated cytosine, then if you treat with bisulfite, all the cytosine can be changed into uracil, and methylated site can stay as a cytosine. Now, if you have one specifically interested gene, you can clone that gene's 5' area. Then, that 5' area has a lot of CPG collected together. Those are what we call CPG island, right? And then, those islands you treat with a bisulfite. If that CPG site is methylated and it is protected, that's why it will give you as original CPG sequence, and then this will be methylated site. If that site was unmethylated, then the C is changed into U, and if you change that into DNA, U changed into T, then this sequence will give you TG, then this is noted by open circle here. Then you can see here, the black circle means methylated site, and open circle means unmethylated site. So after cloning your interested gene, you can sequence them, and then you can figure out which original site of a CPG was methylated or unmethylated. If you have a specific interest site for the methylation, you can utilize methylation-specific PCR. After treatment of a bisulfite, depending on those methylation pattern, the product sequence can be different. And utilizing this difference of sequences, you can design methylation-specific primer for the PCR. If this primer is matching to the methylation pattern, then only this primer can generate the product when your original sequence was methylated. Because this primer cannot match to this sequence, unmethylated gene cannot generate the PCR product. By checking whether this PCR experiment can generate the product or not, you can decide whether this cytosine site was methylated or not. This method can be quite useful if you know exact site of your interest of methylations. If we need to know the whole genomic scale of methylation patterning, we may need different approaches. In this case, we can utilize methylated DNA immunoprecipitation. Immunoprecipitation sounds like we need to use antibody. In this case, we utilize the antibody for methylated cytosine. So if you can get that kind of antibody which can recognize selectively for those methylated site of cytosine, we can take a whole genomic DNA and then chop off. Utilizing this antibody, we can collect only methylated fragment. And then using this fragment, now one way is we can do direct sequencing. So we have learned a lot of advanced sequencing technique for DNA. This is one way. The other way is we can analyze those sequences using DNA microarray. In DNA microarray approach, you can use different tricks. Maybe you can use a two-color system. One color can be for whole genome sequences. The other color for only methylated fragment sequences. If you compare those two, easily you can map out along line the whole genomic sequences, which site is methylated or which site is unmethylated. Now, let's compare with the DNA sequences. DNA sequence has 4 different building blocks, A, T, C, G. So that's why we said those sequences are quaternary systems. Methylation pattern, how about it? That's either methylated or unmethylated. That's a 1 or 0. So that's binary systems. Along line the whole genomic sequences, it easily can be mapped out whether methylated or unmethylated in binary digital informations.